This lecture is on the molecules of life. In this lecture, we're going to zoom in from the cellular level down to the molecular level and look at the chemical building blocks that make life possible. We'll focus on how atoms bond together to form molecules, how those molecules are organized into macromolecules, and how structure is directly tied to biological function. By the end of this lecture, you should be able to understand how carbohydrates, lipids, proteins, and nucleic acids are built, how they differ from one another, and why their chemical properties matter for everything cells do. When we look at a cell, most of it is water, but what really gives us life is the structure and functions are carbon-based molecules. These are known as organic compounds, and they form the chemical foundation of living systems. Carbon is essentially important because it can form stable bonds with many other atoms, allowing cells to build large complex molecules. These molecules are not random. They are precisely arranged to support everything a cell does, from storing energy to building structures and carrying out chemical reactions. So, when we talk about chemistry of life, we're really talking about how carbon based molecules make life possible at the molecular level. Carbon is uniquely suited to serve as the backbone of biological molecules because it can form four covalent bonds with other atoms. Their bond's capacity allows carbon to build a wide variety of structures, from simple molecules to large and more complex ones. Carbon atoms can bond to one another in chains, rings, or branches structures, creating a tremendous molecular diversity. In living organisms, carbon most commonly bonds with hydrogen, oxygen, and nitrogen, forming the basic framework for organic molecules. Even very simple compounds like methane show how carbon's bonding flexibility sets the stage for much larger and more complex biological molecules. Carbon skeletons can vary in length, shape, and branching systems, and these differences matter biologically. A single carbon chain behaves differently from a branched or ring-shaped one, even if the molecules contain the same atom. These structural variations influence how molecules interact with each other inside the cell. Some small changes in the arrangement of carbon atoms can lead to major differences in function, which is why structural diversity is so important in biological chemistry. Methane is one of the simplest organic compounds, yet it can be represented in several different ways. A structural formula shows how atoms are connected, while a molecular formula summarizes the types and number of atoms present. A space filling or three-dimensional model helps visualize how the molecule occupies space. Each representation emphasizes different aspects of the same molecule, and together they help us better understand molecular structures and bonding. Hydrocarbons are organic molecules made entirely of carbon and hydrogen. The energy stored in their bonds can be released during chemical reactions, which is why hydrocarbons are effective fuels. Large hydrocarbons make up gasoline and other fossil fuels, while smaller hydrocarbons and related molecules are used by cells as energy sources. When these molecules are broken down, the energy released can be captured and used to power cellular processes. This makes hydrocarbons an important link between chemical structure and biological energy use. The specific proteins of an organic molecule depends not just on its carbon skeleton, but also on the functional groups attached to it. Functional groups are small clusters of atoms that participate directly in chemical reactions. They influence how molecules interact, how soluble they are in water, and how they behave in biological systems. Many biological molecules contain multiple functional groups, which gives them a wide range of chemical behaviors. Many of the molecules that make life possible are extremely large at the molecular scale. These molecules are known as macromolecules, and they are responsible for the structure and function of cells. Despite their size, macromolecules are built from much smaller components that are assembled in an organized way. Living systems rely on just a few major categories of macromolecules, each specialized for a particular biological role. These are the four major classes of macromolecules found in living organisms: carbohydrates, lipids, proteins, and nucleic acids. Each group has a distinct structure and function within the cell. Carbohydrates are primarily involved in energy storage and structural support. Lipids play a key role in membrane and long-term energy storage. Proteins carry out most cellular work, and nucleic acids store and transmit genetic information. Most macromolecules are polymers, meaning they are made up of many repeating smaller units called monomers. The specific monomer and how they are arranged determines the properties of the polymer. This modular design allows cells to build a wide range of molecules using a limited set of building blocks. By changing the sequence and number of monomers, cells can fine-tune the structure and function of the molecules. Cells build polymers through dehydration reactions, which link monomers together while releasing a molecule of water. This process requires energy and is tightly regulated by enzyme. The reverse process, called hydrolysis, breaks polymers apart by adding water. Hydrolysis allows cells to digest food molecules and recycle macromolecules when they are no longer needed. Carbohydrates include sugars and polymers made from sugars, and they are the main source of energy for living organisms. In animals, carbohydrates provide quick energy and serve as raw materials for building other molecules. In plants, carbohydrates also play a major role in structure, especially in cell walls. These molecules are central to metabolism and energy flow within the ecosystem. Their structure directly relates to how they are used by the cell. Monosaccharides are the simplest carbohydrates and serve as the monomers for more complex sugars. They cannot be broken down into smaller carbohydrate units. Common examples include glucose, which is a major energy source for cells, and fructose, which is commonly found in fruits. Even though these sugars may share the same molecular formula, their atoms are arranged differently, which affects how they behave chemically. These simple sugars are central to cellular metabolism and energy production. Glucose and fructose are isomers, which means they have the same chemical formula, C6H12O6, but their atoms are rearranged differently. That difference is in the arrangement is what gives them different properties. In glucose, the carbonyl group is located on the first carbon of the chain, while the fructose, the carbonyl group, is on the second carbon. The rest of the carbon atoms are rearranged in a similar way, and hydroxyl groups and hydrogen is attached. Even though the formulas are identical, this small shift in where the carbonyl group sits changes how each molecule behaves in cells and how enzymes interact with them. Monosaccharides, especially glucose, are the primary fuel for cellular work. Cells break down glucose during cellular respiration to release energy needed for growth, maintenance, and reproduction. In aqueous environments like the cell, many monosaccharides form ring-shaped structures which influence how they interact with enzymes. All carbohydrates, whether simple or complex, are ultimately built from the basic sugar units. In water, glucose commonly forms a ring rather than remaining in a straight chain form. This ring structure is important because it affects how glucose fits into enzymes and how it links to other sugars. The spatial arrangement of atoms in the ring determines how glucose participates in chemical reactions. This structure helps explain how glucose can be used to build larger carbohydrates like starches and glycogen. The formation of disaccharide illustrates how smaller molecules changes can influence diet and health. Sucrose, for example, is a primary sugar transported in plant sap and is a major component of table sugar. High fructose corn syrup is produced by converting glucose into fructose, increasing sweetness without increasing total sugar content. Understanding how these sugars are formed helps explain why different carbohydrates affect the body differently. Polysaccharides are complex carbohydrates made from long chains of monosaccharides linked together. These molecules are used for energy storage and structural support, depending on the organism. Because they are large, polysaccharides are not sweet and do not dissolve easily in water, like simple sugars. Their size and structure makes them efficient in storing energy over long periods of time. The function of polysaccharides depends on how the sugar units are arranged and bonded. Starch is a polysaccharide used by plants to store energy. It is composed entirely of glucose monomers linked together in a long chain. When animals eat plant-based foods, enzymes break down starch into glucose, which can be used for energy. This makes starch an important connection between plant energy storage and animal metabolism. Starch, glycogen, and cellulose are the polysaccharides made from glucose, but their structure and function are very different. Starch is made of long chains of glucose and is used by plants to store energy, which is why starch granules are abundant in tissues like potato gloopers. Glycogen is in the animal storage from glucose and has a highly branched structure, allowing it to be broken down quickly when muscle energy is needed. Cellulose, in contrast, is used for structures rather than energy storage. Its cellulose chains run parallel to one another and are held together by hydrogen bonds formed strong fibers that can give plant cell wall their strength. Glycogen is the main energy storage polysaccharide in animals. It is stored in the liver and muscles and can be broken down into glucose when energy is needed. Cellulose, in contrast, is structural polysaccharide found in plant cell wall. Although cellulose is made up of glucose, animals cannot digest it because they lack the necessary enzyme to help break it down. Lipids are a diverse group of molecules that do not mix well with water. Unlike carbohydrates, lipids are hydrophobic and tend to cluster together away from aqueous environments. Lipids are not true polymers and are not broken from repeating subunits. They serve important roles in energy storage, insulation, cushioning, and membrane structures. Their chemical properties make them especially useful in forming biological barriers. Most carbohydrates are hydrophilic, meaning they dissolve easily in water. Lipids, on the other hand, are hydrophobic and separate from water. This difference explains why oils and water form distinct layers. In cells, this property is essential for forming membranes that separate inside the cell from the external environment. Molecular interactions with water strongly influence biological organization. The typical fat, also known as a triglyceride, is made up of glycerol molecules bonded to three fatty acids. These bonds are formed through dehydration reactions. Triglycerides are efficient energy storage molecules because they contain many high energy bonds. In the human body, fats also provide insulation and help protect organs. Their structure allows large amounts of energy to be stored in a compact form. A triglyceride is built by joining one glycerol molecule with three fatty acid molecules. Each acid attaches to a glycerol through a dehydration reaction where a molecule of water is released as the bond forms. The result is a compact molecule with a glycerol backbone and three long hydrocarbon tails. These tails are rich in energy because of the many carbon-hydrogen bonds they contain. This structure explains why triglycerides are such effective long-term storage molecules and why they are stored efficiently in adipose tissue for insulation, cushioning, and fuel when needed. When we compare different fats, the proportion of saturated and unsaturated fatty acids really matter. Corn oil is mostly unsaturated, with all about half of its fat being polysaturated and over a third monosaturated, which helps explain why it stays liquid at room temperature. Palm oil is more evenly split, but it contains a much higher proportion of saturated fats, which makes it more solid and gives it different biological effects. Butter contains the highest percentage of saturated fats, which is with very little polysaturated fats, which is why it is solid at room temperature. These differences in fatty acids composition directly influences both the physical properties of fats and their impact on human health. Most animal fats contain a high proportion of saturated fatty acids. Because these fatty acids are straight, they can pack tightly together, which is why many animal fats are solid at room temperature. Diets high in saturated fats are associated with atherosclerosis, a condition where fatty deposits build up in the blood vessels and restrict blood flow. In contrast, most plants and fish fats contain higher levels of unsaturated fatty acids. These fats tend to bend in their structure, preventing tightly packaging and keeping them liquid at room temperature. Hydrogenation is a chemical process that adds hydrogen to unsaturated fats. This converts them into more saturated forms, making liquid oils solid at room temperature. During this process, trans fats can be produced, which is particularly harmful to health risks because they disrupt normal lipid metabolism and increase the risk of cardiovascular disease. Steroids are lipids with very different structures from fats. Instead of long fatty acid chains, steroids have a carbon skeleton made of four fused rings. Cholesterol is an important steroid that helps maintain cell membrane structure. It also serves as a starting material for other steroids, including hormones like estrogen and testosterone. Small changes to its core structure result in very different biological functions. Small chemical differences added to the steroid ring creates molecules with very different functions. Hormones like estrogen and testosterone regulate reproduction. development, and metabolism by binding to specific target cells. Synthetic anabolic steroids are modified versions of testosterone and enhance muscle growth and recovery. While these can be prescribed for certain medical conditions, their misuse can disrupt normal hormone balance and lead to serious physical and mental health problems. Synthetic anabolic steroids are modified forms of testosterone that are designed to mimic some of its effects in the body, especially those related to muscle growth. In medical settings, these compounds can be prescribed to help treat conditions such as cancers or AIDS, where patients experience severe muscle loss. Outside of medicine, anabolic steroids are sometimes abused by athletes to increase muscle size and strength more rapidly than it would be possible naturally. This misuse can disrupt normal hormone balance and lead to serious physical and mental health problems. Proteins are large macromolecules made up of long chains of amino acids and account for more than half of the dry weight of most cells. They are involved in nearly every process that keeps cell alive, including chemical reactions, structural support, transport, signaling, and defense. Because proteins carry out so many different tasks, they come in wide varieties of shapes and sizes. The specific function of a protein depends on its unique structure, which is carefully built and maintained by the cell. Proteins can be grouped based on what they do in the cell. Some proteins act as enzymes that speed up chemical reactions, while others provide structure, such as those found in the muscles and in the cytoskeleton. Transport proteins move substances across membranes or through the bloodstream, and signaling proteins help cells communicate with one another. Defensive proteins, like antibodies, protect the body from pathogens. Together, these diverse proteins functions life makes life the cellular process possible. Proteins are built from smaller subunits called amino acids, and all of the amino acids share the same basic structure. Each one has a central carbon atom bonded to four components: an amine group, a carboxyl group, a hydrogen group, and a side chain. The amino acid group and the carboxyl group are the same in every amino acid, which allows them to link together to form proteins. What makes them amino acids different is the side chains, also called the R groups. These side chains vary in size, shape, and chemical properties, and those differences are what ultimately determines how a protein folds and functions. Leucine is an example of an amino acid with a hydrophobic side chain. It has a standard amino group, carboxyl group, and a hydrogen attached to the central atom, plus a side chain made up of mostly carbon and hydrogen. This side chain concludes CH2 groups connected to a branch carbon that is bonded to two CH3 groups. Because it lacks oxygen or charged atoms, this side chain does not interact well with others. Hydrophobic amino acids like leucine tend to cluster together inside proteins, help stabilize the protein's three-dimensional structure by avoiding contact with water. Protein function is directly tied to protein shape, and that shape begins with how amino acids are linked together. Amino acids are joined by a peptide bond, which forms between the carbon atom in the carboxyl group of the one amino acid and the nitrogen atom in the amino group to the next. This bond forms through a dehydration reaction, releasing a molecule of water as the two amino acids are linked. This peptide bond forms repeatedly. A long chain called a polypeptide is created. This specific sequence of amino acids in what chain determines how the protein folds, and what fold is what allows the protein to perform its specific biological function. Amino acids are joined together through a dehydration reaction that forms a peptide bond. During this process, the carboxyl group of one amino acid reacts with the amino group of the other, releasing a water molecule. The resulting peptide bond links the two amino acid groups into a joining chain. As more amino acids are added, this chain becomes a polypeptide. The order of which amino acids are joined is critical because it determines how the polypeptide will fold into a functional protein. Proteins are built using just 20 different amino acids. Those amino acids can be arranged in chains that are hundreds or even thousands of units long. The exact sequence of amino acids acts like a set of instructions that determines how the proteins fold into three-dimensional shapes. That shape is what allows the protein to form a specific job, usually by recognizing and binding to another molecule. Even a smaller change in the amino acid sequence can disrupt folding of the function. A classic example is hemoglobin, where Replacing just one amino acid alters the protein shape and links to sickle cell disease. Here we see how long and specific protein sequences can be. The polypeptide may contain well over 100 amino acids arranged in a precise order, such as the beginning of lysine, valine, and phenylalanine, glycine, and arginine, with alanine appearing later in the sequence. Each amino acid, like alanine, has the same basic structure, but the different side chains which influence how the chain behaves. As the polypeptide forms, it does not stay flat. Instead, it folds into a three-dimensional shape. Ribbon models highlights this folding, showing coils, helices, and folded regions, all of which contribute to the final structure. This complex folding pattern is what allows a protein to function properly inside of a cell. A very small change in a protein's structure can have major consequences. If a protein folds into the wrong shape, it can and may no longer function properly. And this loss of function can lead to diseases. Many disorders, including several serious nervous system diseases, are linked to proteins that have misfolded. Protein shape is also highly sensitive to the environment inside of a cell. Changes in temperature, pH, or other conditions can cause proteins to unravel, which disrupts the structure and prevents it from doing its job. Proteins must fold into very specific shapes to function correctly, especially in the nervous system where precision is critical. When a protein folds improperly, it may fail to perform its normal role or begin interacting with other proteins in harmful ways. In some cases, misfolded proteins clump together, forming aggregates that interfere with normal cell functions. Neurons are particularly vulnerable to the kind of damage because they are not easily regenerated. Over time, the buildup of misfolded proteins can disrupt communication between neurons and lead to progressive brain diseases. Most of these are caused by prions. Nucleic acids are macromolecules that store, transmit, and help express genetic information in cells. There's two types, DNA and RNA. DNA holds the long-term genetic instructions that determines the organism's trait and guides cell function. RNA acts as a working copy that information and plays a direct role in building proteins. Together, nucleic acids connect genetic information to cellular structure and function. Without nucleic acids, cells would not be able to maintain their identity, reproduce, or make the proteins necessary for life. Nucleic acids are the molecules that store and transmit genetic information. In humans and all other organisms, this genetic material is DNA. DNA is an extremely large molecule, and inside the cell exists as a long fiber of chromosomes. Each chromosome contains many genes. A gene is a specific segment of DNA that carries instructions for building proteins by determining the amino acid sequence of the polypeptide. In this way, nucleic acids link inherited genetic information to structure and function of proteins that keep cells and organisms alive. Building a protein begins with the information stored in the DNA. That information is transcribed into RNA, which then carries the instructions to the cellular structures that assemble amino acids into polypeptides. This sequence of nucleotides and nucleic acids ultimately determines the sequence of amino acids in the protein. This flow of information explains how genetic instructions are transmitted into functional molecules that perform work inside the cell. We start from the DNA, we make the RNA, and that RNA is what helps make the proteins. Nucleic acids are built as long polymers made from repeating subunits called nucleotides. Each nucleotide has three basic components that work together to form the structure of DNA and RNA. There is a 5-carbon sugar which provides part of the backbone, a phosphate group which links nucleotides together, and a nitrogen-containing base which carries the genetic information. By arranging these nucleotides in specific sequences, cells are able to store and copy, transmit genetic instructions with remarkable accuracy. A DNA nucleotide is made up of 3 connected parts, a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base. The phosphate group links one nucleotide to the next, forming a continuous chain. Deoxyribose acts as the central connector, attaching to the phosphate on one side and the nitrogenous base on the other. In this example, the base showed is thymine, and one of the 4 bases found in DNA. While the chemical details are complex, the key idea is that repeating nucleotides connect through sugar-phosphate bonds to form the backbone of DNA, with the bases projecting outwards to carry genetic information. DNA contains four different nitrogenous bases, and they fall into two structural categories. Adenine and guanine are larger bases made up of two fused rings, while thymine and cytosine are smaller bases with a single ring. Adenine bonds with thymine, and guanine bonds with cytosine. Even though their detailed chemical structures differ, the key idea is that these bases have specific shapes and bonding patterns. These patterns allow them to pair in very specific ways inside the DNA molecule. DNA is formed when nucleotide monomers are linked together through dehydration reactions. These reactions create a covalent bond between the sugar of one nucleotide and the phosphate of the next, producing a repeated sugar-phosphate backbone. The nitrogenous bases extends outwards form the backbone and carry the genetic information. In cells, DNA exists as double-stranded molecules with two polynucleotide chains twisted into a double helix. The bases on one strand paired with the bases on the other through hydrogen bonds, with adenine pairing only with thymine and guanine pairing only with cytosine. The specific base pairing is essential for DNA stability and accurate replication. DNA and RNA are both nucleic acids made from chains of nucleotides, and each nucleotide contains a sugar, a phosphate group, and a nitrogenous base. Despite these limitations, there are important differences between the two molecules. DNA contains the sugar deoxyribose, while RNA contains ribose, which has one additional oxygen atom. DNA uses the base thymine, whereas RNA uses uracil instead. Structurally, DNA is typically double-stranded and forms a double helix, while RNA is usually single-stranded. An RNA nucleotide is composed of three parts: a phosphate group, a ribose sugar, and a nitrogenous base, which in this case is uracil. The phosphate group links nucleotides together to form the backbone of RNA strand. Ribose is a 5-carbon sugar that differs from deoxyribose by having an extra hydroxyl group, which makes RNA more reactive and less stable than DNA. Uracil is a single ringed base that replaces thymine in RNA and pairs with adenine during base pairing. These structural features supports RNA's role as a temporary molecule involved in gene expression and protein synthesis.